I'm the senior partner, the senior technical partner to the university, so I'm the conduit between DSTL and Southampton. So feel free to get in touch and I'll try and put you in contact with the right people. This is just an overview of DSTL, so I'm watching for cringing if I say something wrong here. Oh, I'm going to have to come and press there, sorry. So, yeah, just a very brief overview of DSTL. So we've got about 4,500 members of staff, currently over 6 sites. Those sites are being rationalised, that we've got 2 main sites. The one that most of you heard about is Porton Down. The other one's Portsdown West. And then we will have then a satellite site at Alverstoke as well. We have just over £700 million worth of turnover. We are part of the Ministry of Defence. £321 million worth of that goes out to industry. And what we do is just in this little diagram here. So we do operational support to the military. We have expertise that's been developed at DSTL over a very long time. And that's been brought to bear over the past couple of years, and then before that, the incident in Salisbury. Then we look at requirements from our military stakeholders and customers, and then we evaluate solutions. So we're the independent part of the Ministry of Defence that looks at those different solutions that people come to us with. And then we do our own research as well. And what we're looking to be is the science inside the Ministry of Defence, and we're looking to deliver high impact S&T. I keep pressing this, I'm going to have to do that again. So we have recently reorganised how the research is done at DSTL. We have these 22 strategic capabilities. These are the big handfuls of the research that we do. These are in a different colour at the bottom just because they're the systems areas. For this meeting, these are the ones that I've highlighted would be interested in the microbial impacts. So we've got CBR, which is our home division. We've got human and social sciences, so that's the microbiome work generally. But then if we think about above water systems and underwater systems, there's definite uses there, biofilms, encouraging them, or various coatings to discourage their growth on the hulls of ships and things like that. It's really broad, all the work we do. And then of course advanced materials as well. So a little bit of more detail about what we do. So we advise and support different operations. You can see here there's images of the work in Salisbury after the incident there with the nerve agent. We understand and anticipate threats, so there are specific teams that look at all of the different research that's going on around the world, try to understand what people are looking into, what could be used as a threat against us, and then how do we start to approach working against that. As I said before, we test and evaluate. Improve medical outcomes, so we have a team in our division that specifically looks at trauma and the immediate treatment of people who have had traumatic injury. Part of us being here is to deter our adversaries as well. And then finally, it's to develop novel capabilities. And we're just going through a mandate for the next 5 years and reformulating the different pieces of work that needs to be done. So now's a really good time to come and start talking to us. The division that we're in, CBR, these are the main areas of work that we do. So modelling of different scenarios out in the environment, as well as hazard assessment and advice on how to respond to events like this. Physical protection, so images there, pretty obvious, people wearing respirators to protect their respiratory system from threat agents, but then we also have collective protection, which are shelters which provide that protection to whole groups of people without them needing to be in those respirators. We look at medical countermeasures, and that can be from the concept phase, looking at new things. Adam indicated this in his presentation earlier, right through to taking things to licensure. We work on detection and diagnosis, and Petra mentioned that earlier. I've got a slide at the very end of my presentation about the synthetic biology themes. And then, of course, we look at decontamination as well. So any of the work that you're doing, think about these areas and where that work could be applied. The group which is most pertinent to this meeting is our Microbiology and Aerosol Sciences group. And we have a state-of-the-art facility at Porton Down where we can do containment up to level 4. Those are experimental laboratories as well as animal laboratories. And here's just a couple of examples. So we have the non-human primate marmosets. And then, I can't remember who it was earlier, as well mentioned insects. So these are wax moth larvae, Galeria melanella. That's a good species to be able to screen different approaches to microbial protection. We have these key enablers that allow us to do this work. Clearly we're working with some very hazardous pathogens, and being able to handle those safely is key. We're able to test and evaluate in realistic environments. So what we can do is we can use simulants out on our range. We've got a huge range up at Porton Down where releases can be done using simulants. We obviously wouldn't use live agent there, but we can look at how these things would move in the environment, and that obviously informs some of that modelling. We can work with animals. I mentioned the marmoset. We have several different species that we can work with in containment, and that's certainly one of very few facilities around the world that can go right up to category 4. And we have that regulatory knowledge from our advanced development programme, where we're talking to the regulator and getting things right through to market. So our customers and stakeholders, unfortunately in the email across to the university, I've had to compress all these pictures. That's the Counter-Proliferation and Arms Control Centre. Those are Ministry of Defence stakeholders that we work with regularly. Then we work with other parts of government, Home Office, Centre for Protection of National Infrastructure, and then we work internationally as well with our counterparts in primarily the US, but we work the world over. So the medical countermeasures projects that the group tends to work on has 4 main elements. 2 of them, the target antigen discovery and the efficacy evaluation, are done in DSTL. That's where that work has to be done because of what we're working with. But otherwise, we partner with external groups and external organisations to look at the drug identification and then clinical trials as well. So I'm just going to take you through each one very briefly. So target and antigen discovery, we've got a vaccine programme that's looking at these different agents, and you can see here, I'm not going to read through them, but we've got various different vaccines in the programme, and we're working with all of these different partner organisations to deliver this. The requirement for us to go this alone is too big, so we burden share with our partners in the US, Canada, Australia primarily, but with others as well. Then we also have identification of post-exposure therapy, so this is after a person's been infected, and what we're looking for here is to identify targets, and then we can down select those if we've got a hit, and then once we've got a hit we know what we're trying to work against, be it a particular receptor or a particular pathway, then we can look to identify some lead compounds and take that through. I'm aware that this is very much antimicrobial at the moment. And then we would take that through into our efficacy evaluation, so that's our animal models in containment. Then we've got the enhancing current medical countermeasures. This is the project that Adam presented earlier in his very quick slot. So this is where we're looking at antimicrobials that are already in market, looking at how we can reformulate them or deliver them differently to be able to give protection against our threat agents. And so this is the approach we're using. So we're looking at alternative indications, so looking at those drugs that are already out there? Can we look at other indications and get the license changed? Can we look at novel therapeutics? So this is your lower technology readiness levels. This is that early novel compounds or approaches. We can then look at formulations, and again, Adam mentioned this, working with different people to look at how we can encapsulate and deliver these drugs which normally be given by the parenteral route as inhaled agents and try to affect the site of infection. And then also combination therapies. What else can we give to boost the protection and the survival against these agents? And of course, we don't do all of this alone. As Adam said, we're not a drugs company. We're not into discovery. What we're trying to do is pull through. And then finally, on this slide, models for comparison. So these are the different approaches that we tend to use. This is the pathway. So we'll start with in silico modeling or advanced in vitro models, organ on a chip, lab on a chip kind of approaches. Then we'll go into our proof of principle species, which tends to be the mouse, but we can also do that Galleria insect model. If something is looking good, that's when we then go over into the non-human primate. And if that pivotal efficacy and all of the pipeline, that pharmaceutical pipeline looks good, then we'll move through into licensure. Just to highlight with the marmoset, which is, like I said, that's a unique capability that we've got. We're able to look at all of these different pathogens in that model in containment, and so that's something that if the work that you guys do or the work that we're doing pulls through into that, and that then becomes that pivotal evidence for licensure. So then, yeah, just in summary, we can work with bacteria and viruses up to level 4, and we can do that operationally or an emergency response. We can generate aerosols of both the therapeutics as well as those pathogens and deliver them to the lung of the animals. We can do genetic manipulation of the pathogens, and then we can do the downstream analysis as well, so immunology, molecular biology and imaging. Slightly changing tack here, we are also interested in the microbiome. We have currently got a couple of PhDs ongoing, one's here at Southampton, the other one's with the Quadram Institute, and that's looking at the gut microbiome in both those cases. But we're not only interested in the gut microbiome. David earlier spoke about the respiratory biome, and we've had talks about skin as well. So this is generally in the area of human performance or augmentation. So if someone becomes ill, what happens to the microbiome? Can we do something through the microbiome to improve their health? And that's a whole big new area of work that's ongoing. There is a lot of work in this area already. So partially what we're doing is just looking at the data that comes out of that and being aware of it, but we're more than happy to engage and look to see what research we can affect in this area. And then my final slide is just what Petra presented very, very briefly before. These are the challenges in synthetic biology, which some of you guys might be interested in. So we're interested in power, materials and sensing. And so what we've got here are the actual challenges, examples of those. So novel non-batteries, disposal, that's a terrible typo, recycling and remediation, feedstocks and fuels. And then, with those, how could that benefit the military? So we're looking at place and forget technology, sensors that you leave behind, they don't need a battery, and they're collecting that data. Biogeneration, so if our armed forces go out into theatre, how can we get them out there with the power that they need without the huge logistic burden of taking everything that we need with us? Can it be generated in situ? And then, of course, waste into power. Again, that's something that can benefit us and reduce that logistical burden. Anyway, Petra very briefly had her poster there. if anybody's interested in these, they will be getting posted out via our cloud, which is our commercial framework, and we'll be having webinars on those. So, you know, make sure that you register to hear about those, so that when they come up, you can join the webinar and find out a little bit more and see whether, you know, that's an area that we can support your research, and you can give us solutions as well. And that's me.